This is Clive Wearing and his wife, Deborah. Clive is a former musicologist, conductor, tenor, and keyboardist that suffers from chronic anterograde amnesia as well as retrograde amnesia. This means he cannot store new memories or recall past stored memories. He lives his life now constantly believing he's just woken up from a coma, completely stuck in the present. Clive still remembers how to play the piano and conduct his choir, even though he cannot remember any of his musical education. Because of this, scientists knew that his procedural memory was still intact, and his cerebellum, where procedural memory is stored, wasn't the issue. Clive was constantly keeping a diary, writing down every time that he felt like he'd woken up from his state of coma. You could see his emotions getting stronger and stronger as he got frustrated and crossed out the line before it, insisting that this is the time that he actually was awake. He was officially named the man with the 7-second memory. Although Clive's cerebellum had remained completely intact, it wasn't the same case for his hippocampus. Clive had fallen ill to a rare form of herpes encephalitis virus. In this rare case, the virus had attacked his brain and left Clive's hippocampus almost completely destroyed. Remarkable enough, as damaged as his hippocampus was, the one person he could remember was his wife Deborah, due to his undeniable love he has for her. The reason why this was so detrimental to Clive's memory is because this is where our brain encodes memory. It is where the outside world is interpreted by our synapses. Our synapses are the points in which our neurons communicate. So this then leads to the question, how do our neurons actually accomplish this? How are we able to not only store and recall past memories, but encode our experiences through neuronal activity? In response to an external event, our brain will either encode sensory memory, short-term memory or working memory, or long-term memory. In order for us to have these type of memories, our synapses of our hippocampus will have to strengthen in order for the memories to be encoded and then stored within other parts of our brain. This is a process called memory consolidation of the hippocampus. Let's look at how this happens. In a neuron, we have dendrites at the top, a nucleus, and then an action potential that runs down the axon until it hits the terminal. At this end, a synapse is formed. This is where the terminal and the dendrites of the next neuron talk by neurotransmitters and receptors. There is an external signal. This may lead to an action potential in a neuron. When neuron 1 releases its signal from the stimulus, neuron 2 will receive it. If this action potential is sent down the axon repeatedly by the repeated stimulus and they fire at the same time, they will form a stronger connection. They call this neurons that fire together, wire together. However, the timing for this interaction is crucial. If the second neuron does not receive the signal soon enough after the initial stimulus, the connection will be very weak, and this will not be a strong synapse for memory consolidation. These strong connections between neurons is the basis of what we call synaptic plasticity, in which we either use it or lose it. These connections are very susceptible to change, as so is our memory. So let's say we have our presynaptic and postsynaptic neurons. The presynaptic neuron will have the neurotransmitters that are released due to the action potential that has been traveling down the axon. These will enter the synapse between the two cells. The postsynaptic cell will have the receptors specific for the neurotransmitter released. The more action potential, the more neurotransmitter will be released, and the more receptors will be put on the postsynaptic membrane. This will then up the neurotransmitter release again, forming the strong connection the cell wants to form this memory. In Clive's brain, there is only a bit of hippocampus left on the left hemisphere and none on the right. This is why Clive processes no more than 7 seconds after an event. Once the event happens, the neurons do not make a connection in the hippocampus, which is why no new memory is encoded or stored within the brain. Because it is difficult to study the memory of a human, scientists are studying the hippocampus in mice. When mice are learning something new in response to a stimulus, the scientists will tag the neurons that are activated. Then, in a different environment at a later time, they'll reactivate these neurons. Scientists are starting to see that this re-stimulation alone leads to the same behavior response without the initial stimulus. So let's see exactly how this works in the postsynaptic cell. There are two types of receptors for memory neurons of the hippocampus. These are the AMPA receptors and the MDMA receptors. Hyperpolarization of the cell leads to the magnesium being lodged in the NMDA receptor, allowing nothing to go through it without the proper environment. Sodium is permeable through the AMPA receptor, while calcium and sodium are both permeable to NMDA. When there is a large amount of action potential from the presynaptic neuron, there will be a large release of glutamate, our memory neurotransmitter. When glutamate binds to AMPA, sodium will then go into the cell. If there is enough glutamate released due to a strong or highly repeated stimulus, and enough sodium is within the postsynaptic cell, the neuron will depolarize due to the rush of sodium that entered through the AMPA receptors. Because of the passing of threshold to depolarize the cell, the magnesium of the NDMA receptor will be pushed out and allow all of the calcium to rush in. Calcium in this environment acts as a second messenger and will start a signal cascade to put more AMPA receptors on the membrane so that the connection can be even stronger between the two neurons. NMDA receptors are considered a coincidence detector. It not only has to detect the glutamate in the synapse from the postsynaptic neuron, but also the threshold of the depolarization from the AMPA receptors on the postsynaptic neuron. This high increase in AMPA receptors on the postsynaptic membrane leads to a strong short-term response. If this process is continued, the response strength will become more long-term. The calcium will signal transcription factor that will encode for growth factor. This increase in growth factor transcription will work to strengthen the synapses, trying to make the synapse connection as strong as it can be. This will form what we call long-term potentiation. This long-term potentiation is what allows our hippocampus to encode more long-term memories. When we look at the communication between neurons, we know that neurons have a lot of dendrites. This means that this can be acting on a lot of different dendrites at once, only increasing the signal. Therefore, the more the connection, and the more action potential we get over a longer period of time, we can get long-term memory. Long-term memory is ultimately formed when these connections don't just happen between two individual neurons, but many. This will build the connection of neurons that is necessary to turn an external stimulus into a memory that we will hold for an indefinite amount of time. Clive Waring is now 81 years old, and he has been living in the present for 31 years. Clive's rare illness and even rarer state of mind has inspired large amounts of scientists to study the hippocampus and how it encodes memory for future storing in the brain. We will just have to wait and see what other crazy things we learn about how these neurons keep our brains so plastic.